So I want to talk a bit about life at a single molecule level, introduce a couple of techniques, the things we've been doing in the lab over the couple of years, and is this supposed to work? Yeah, I have to press down to go. Okay. Okay, great. Oh, yes. I will try to remember. Okay, so first question is, where's Wageningen? And it's somewhere right in the center of the Netherlands. Yeah, so this is the coast of the Netherlands, Wageningen, somewhere here. And university kind of focuses very much on the life science and agriculture and so on and so on. And that's a kind of a bit of an older picture of the campus, new campus with way more buildings coming in here. Also a lot of industry putting now their research and development departments into the onto the campus to have a relatively close connection. Okay, so I would like to start with that picture. That's a bright field image of a couple of bacteria. And as a physicist, you would say, hey, great, spheres. Yeah, first approximation. So a couple of micrometer long, yeah, so 1-2 micrometer in width. And then the biologists will say, well, wait a minute, yeah, it's a bit more complex than a sphere. So if we now dive into a bit, and this is a very nice drawing by Gutzal, published recently, it looks way more complicated. Yeah, a lot of things going on. Yeah, so you see basically all the entire volume is filled with proteins. And I'll pick out a couple of things. Yeah, so here in yellow is kind of the long DNA strand that goes over through the bacteria. You have then a ribosome sitting here, translating the DNA, making new proteins and so on. There's another protein which was actually added only last year by the artist. Yeah, because he had an older version of that was 10 years ago, 10 years older, which didn't include this protein. Yeah, so that's a CRISPR-Cas protein. And for a special kind of those ones, Cas9, there was kind of Nobel Prize in chemistry a couple of years ago. So I will talk about the second half of my talk about Cas proteins. And the breakthrough basically came in the field of people came up with this idea and discovered the use of genetically encodable fluorescent proteins. Yeah, so the idea is that because it's genetically encodable, you can fuse it to any gene of interest and then watch that singular protein in a cell, in a cellular environment. Yeah, so that was the second Nobel Prize, now quite a while ago, to Roger Tsien and others. And that basically opened up really a field, yeah, because you can excite with, let's say, green laser, you excite the fluorescent protein, it will emit light, and you can say where it is in a cell, how it's moving, and so on. And the big problem is here that even though we have a lot of structural features available of most or many of the proteins, what we do not really know is what is actually the dynamic interactions between the proteins and how do they work together in a cell. Yeah, because the picture, of course, just gives a static picture. We don't know much about the dynamics. And our idea was a bit then, okay, let's have a look into the dynamics. What can we learn and how can we do that? Okay, so the next issue is that if you have this very nice fluorescent protein, looks nice and small, but what you actually detect on the camera is something like that. Yeah, so that's what we call a point spread function. That basically means that instead of seeing something confined in a few nanometer, we'll get a blob. Yeah, and I like the analogy with the blobology in a way. Yeah, so we also have kind of blobology going on by basically having something which is almost in the dimension of the cell. Yeah, so the question again is, can we get around that? That's the third Nobel Prize, which will play a role in my talk today, to Mörner, Betzig, and Hell. How to go from this blob back to the dimension of the fluorescent protein. Yeah, so let's combine all them. And kind of as the rough outline of my talk is, I will talk a bit, hopefully make a case for open microscopy, talk a bit single molecule localization microscopy. What is it? How to do that? How can we improve it? Tell you a bit about technical advancements that we have done over the last years, before then talking a bit about this concept of DNA target search in live bacteria. So again, the point is always we have a fluorophore, we excite the fluorophore with a laser, and then we somewhere redshifted, we detect emission in a different band. Okay, so let's start with a protein of interest. This is labeled order roughly 10 nanometers. What we see on the camera is this plop, this distribution of intensities. So meaning if we, for example, do a histogram of those intensities and the X and the Y, this on the camera represents roughly 500 by 500 nanometers. So we lose an order of magnitude, but we know what to expect. We know the noise to expect. We know what the center is. And then we can fit the intensity distribution with a two-dimensional Gaussian to determine the position of that molecule with relatively high precision and accuracy. Yeah, so precision we can achieve depending on how many photons you have, the more the better, roughly 20 nanometer localization precision. Yeah, and this is great because that brings us close to the actual dimension of the protein. Setups, you will know, simple. Yeah, so you have a laser, focusing the laser light with an objective into your sample, and from that sample, then you detect fluorescence emission on a camera. Yeah. And what's now the idea about super resolution microscopy? It's relatively simple. So the issue we have is that we have, let's say we have a structure, we put a couple of fluorescent labels around, but we are limited by this diffraction limit of half a wavelength. So in our case, roughly 250 nanometers. And how now to do super resolution? I indicated it partially in the last slide. So let's say you have many, many fluorophores. You cannot discern them, but you have to find conditions where you see less than one on average being fluorescent. Yeah. So let's say I find conditions where all of them are not fluorescent. Yeah, will be completely dark. And then if they start blinking, and as long as the distance between those is relatively large, I can basically determine the position of that single fluorophore, again with relatively high precision. So if I go on and on and on, yeah, I can eventually resolve the entire structure. Yeah, and generally speaking, the concept is you separate the emitters in time to separate them in space. Yeah, and there are many, many different concepts on how to achieve that, and I will go through a few of them. But that's the general idea behind super resolution microscopy, at least for single molecule localization microscopy, and many different variants. Okay, so I give you a first example. So what's commonly done is that, for example, you have a biological structure, you have antibodies against that structure, you put a fluorophore on those antibodies or use secondary antibodies, and which direction? Then you find conditions with some chemicals where this fluorophore switches between an on and an off state. Yeah? And Torben, for example, has worked for more than a decade on that. What you get is a video like this. Yeah, so this is now microtubuli stained in a cell, labeled with these antibodies, and you see all those molecules blink. So they go on and off and on and off. If you look very closely, you will see that some of them are a bit more round, and some of them are a bit elongated, either in the X or in the Y axis. Yeah, we'll come to that as well. Okay, and the main issue that we have in all this microscopy is always you have to optimize many things. Yeah, so you would like to have a very high spatial resolution. You would also like to have a high temporal resolution. You do want to avoid phototoxicity, yeah, because if you shine your laser too much into your samples, then cells are normally not very happy. You would like to image in depth, and you would like to image as many molecules, bacteria, cells as possible. Together, we had the issue of relatively high cost of instrumentation, limited accessibility, and so on and so on. So we and basically many others started addressing many of those things. Yeah, and I will give you a few examples of what we did. And before I continue, I would like to bring your attention to kind of a comment paper that we published a few weeks ago about open microscopy in the life sciences. And I would like to generalize it a bit more, yeah, because we are now in a conference with many different people from many different fields, because many of the ideas are rather general, yeah. So in our view, what we have in a kind of a closed research environment, you have your research question, you develop your theory, you experiment, and you publish results. And often, a lot of the information possible to repeat experiments and so is relatively confined into a lab, yeah, or into a scientific area. So if I'm now here on the conference, I can chat to people. I can ask, so how did you actually do it? Because I was missing the information in the supplement. But on the other hand, that excludes many people, because many people are not here. They cannot ask you the specifics. They might or might not be scared to drop you an email. You might or might not answer that email. Yeah. So the point that we are making is that you should, or please think about, how can you make your research as open as possible? Yeah, which information do you share? Yeah, for example, that can mean that you share openly design and resources, that you share your software with how you do the data processing. Yeah, you can publish, I mean, most people, I hope, are doing that already, publish your papers first or on a preprint repository, yeah, so that people can interact with you. And I think in the general, for the long run of science, that will be very important, yeah, because you will get a lot of input from others that you might not have reached otherwise, yeah. Okay, now a bit to the science. So what we started with is designing a kind of a relatively cost-effective, simple instrument, where we also did then most of the experiments I will show you later about the miCube, which has been now picked up in different labs. And in fact, many of new publications use this as a starting block to develop their own versions. And that's basically what I would like to have. Yeah, I'll put something out, people are happy with it, they improve it, and they go on. And we had it added here, small add-on that is known as a astigmatic lens, yeah, and the idea is here that you distort your optical imaging in a way that you become sensitive on where your molecule is relative to the focus. Yeah, so the initial point I've shown you, if you're in the focus, you have a round spot, nice, and then depending whether your molecule is above or below the focus, it will elongate in one or the other directions. Yeah, so that is one way of getting three-dimensional spatial resolution on a two-dimensional camera chip. Okay, so now we have this, say we have this data. How do we analyze it? And there were a couple of, a lot of software available, but most of them relatively slow or inaccessible. So we came up with a new idea. And instead of doing a Gauss fit to determine the position of the molecule, a colleague of mine told me basically 10 years ago, hey, you should do something with phasor analysis. And I ignored him for 5 years, which was a big mistake. So what we now do is we do a Fourier transformation, and I will explain how that works. So let's say you have a molecule in a certain region of interest, and the molecule is elongated, so the emission of the molecule is elongated in one direction because we added the cylindrical lens. Yeah, and what the phasor analysis now does is that we take the Fourier transformation of that image, and we get, if we look at the first harmonic, we get two complex numbers. And those complex numbers we can plot in a so-called phasor plot, which runs always from 0 all the way to 2 pi. Yeah, and we get those two complex numbers we can plot, so they give us an angle over here. So this is this one and this one, and they give us a magnitude. Yeah, and if you do Fourier transformation, you know whatever, if you have a long feature in the real space, then the feature is small in the Fourier space. Whereas if you have a relatively small feature in the x direction, so the width, then the feature will be relatively long in the Fourier space. If you now do the inverse Fourier transformation, then you end up with exactly the point, or pretty much the simulated point in your analysis of the position. Yeah. So here you plot basically from 0 to 2 pi, from 0 to 2 pi. So this is the angle. And the magnitude gives you some information about the width. So what's the big advantage of that? It's very fast because it's a computation. It's not a fit. Yeah, in the fit, you basically have a Gaussian function. You wobble around with that function a bit until you have the best overlap. But for the Fourier transformation, you just say flip, and it's a calculation. And it's non-iterative. It's very fast. So we compared it in our method on a CPU, where we are much faster than a GPU, which has the big advantage that we can now give the software, which we implemented in a Thunderstorm ImageJ plugin, to students, let them take data, and they are then able to analyze on their home laptops within a few minutes their data, whereas before they would have taken them hours to localize. Okay, so that's implemented and available. So if you do any super resolution, give it a go. Okay, next thing is a story that we kind of submitted on bioRxiv a few months ago. The idea is, or the question was here, how do you measure color? Yeah, so normally we have black and white sensitive cameras. We do not have a lot of photons. So how do you then determine the color, so the fluorescence? So here again, the problem, yeah, if you do standard kind of microscopy, you have to kind of, and you do want to do multiplexing, then the emission of organic fluorophores or fluorescent proteins is relatively broad. Yeah, so in the standard spectral range between, say, 450, 700 something nanometer, you will have a huge overlap of the emission. So it's very difficult to separate them, or you have to separate them really widely. Gets more complicated if you, for example, say, I would like to detect in the red region because I don't want to have so much background in the blue and the green. So is there a way that, say, I use a single laser to excite 3 different dyes, and then you see the issue, where they spectrally overlap a lot. So what can we do about it? And the idea we had is really simple. So you take a placed transmission grating, and instead of putting it very far away from the chip, you bring it in very close proximity to your camera chip. Yeah, so less than a centimeter. So what will you see? You will see a point. Yeah, the point spec function that kind of gives information on your localization. Yeah, and you see a slightly extended point in the plus first order, which reports basically on the spectrum of the emission. Yeah, so instead of now measuring the spectrum over many pixels, we'll try to condense the spectrum as much as possible into a single point. Because the advantage then is that if we can approximate the first order as a single point spec function, we simply have to measure the distance between those 2 points to tell us something about the spectrum of the emission. Yeah, so by that, you have, in a way, the most photon efficient way of determining the average emission of a fluorophore. Okay, so how does it look in a simulation? Yeah, so we have 0th order, 1st order, a certain distance in between. If the distance is larger, then that tells us, okay, we probably have redshifted emission. This is how the raw data looks, which is pretty okay, I'd say. Yeah, so you always see this kind of pairs popping up. Yeah, so you can use your standard localization software, and you run then a software that we have again open online, which just finds pairs, and that's it. And then you measure the distance between those two localizations, giving you information on the colors. So here's an example. So here we did, again, storm imaging. So we labeled clothrin pits with one fluorophore and microtubules with another fluorophore. It's kind of a super resolved data structure. And then now we color coded from the emission. And if you select certain areas and compare the areas against each other, then you can make a good case that you can distinguish fluorophores that otherwise largely overlap in their emission spectrum with the technique. Another example here is a DNA paint that I think is very famous in Munich especially. Yeah, so you label your structure with a single strand of DNA, then you have a large reservoir of complementary strands in your solution. And by tuning the kind of the overlap between those two strands, every now and then one of those molecules will bind. Yeah, it's localized, you'll have a larger emission, you can localize it, and then it will diffuse away. So we use some latex beads here or glass spheres that we labeled basically with those single strands. That would be our diffraction limited spot. And then now we super resolve that with DNA paint and use the multiplexing with the spectral resolved imaging to then being able to distinguish basically three different strands, three different spheres here. Okay, the last application for the spectral one is in single molecule FRET. Yeah, so what people are there interested, they're interested in measuring the distance between, say, a donor and an acceptor dye. And what you see here is kind of the emission as a function of the wavelength. And this is kind of the dotted line is the known spectrum of your dye. And the solid line is the one if we deconvolve all our characteristics of quantum yield of the camera and the filters that we use. Yeah, so we cut into that spectrum a bit. And again, the closer then you have an acceptor approaching the donor, the higher the energy transfer will be from the donor to the acceptor. Yeah, so in the case of 55% FRET, that means that roughly half of the intensity you would normally measure in a green or red channel. But that means that if I look in the spectrum, I have a relatively wide spectrum, basically with 2 points. Yeah, in our case, we are not even interested in seeing 2 points. We just look at the position of this one, medium position, and kind of the width. So that allows us to have an additional readout. Yeah, so we can use a single camera chip. We don't have to split in different channels. We'll just measure the distance between the 0th and the first order. Here's one example of a 55% FRET sample where we measure that in time. Yeah, so with a 100 millisecond time resolution. And you can plot the distance as a function of time, yeah, which decreases over here. And you have an additional readout because you can also plot the width of the spectrum as a function of time. And you clearly see here one of those cases where the distance and the width decreases. This is because the acceptor blinks, yeah, so meaning it's not photoactive for a while, cannot receive any energy from the donor. And that shows us you basically that you have a good chance of even doing single molecule FRET, yeah, with the spectral resolved imaging. The big advantage is now you can do multiplexing as you want. Yeah? So you can, in theory, shine a couple of different laser lights onto your samples. You don't need, as long as you can block the laser light, you don't need any additional components to do kind of the spectral measurement. Okay, how I am doing in time? 15 minutes. Okay, now I can slow down a bit. Okay, so the second part of the talk is then about this CRISPR-Cas. So what is it? Basically, the Nobel Prize in chemistry went to two women and for the development and discovery of Cas9. So Cas9 is a protein that you can load with a guide RNA. It's that molecule. And then that protein will find the complementary sequence to that guide RNA somewhere in the chromosome. Yeah, so The guide RNA is normally around 20 base pairs long. Yeah, so if you have, you can find a specific complementarity to whatever region you want on the DNA. Yeah, so you can target a certain site, and then by adding additional DNA molecules that are partially complementary to the left and the right side of that, you can put in a modification at a certain site. Yeah, so for example, you can add a new gene, or you can change DNA. Yeah, so whenever you want to repair a DNA strand or something, then CRISPR-Cas is the technique to find this specific site and do kind of an interaction with that. Yeah, so for genomic editing, that's basically a really huge thing. Yeah, because it's super efficient, it's very simple. You just need the enzyme and whatever guide RNA to find a certain sequence. If we now look a bit into biology and the origins, it kind of started as a curiosity, yeah? Because bioinformaticians found that there is some sequence complementarity between bacterial genome and viruses. And then people started wondering, okay, how can that happen? Why does some of the genetic information of the virus ends up in the chromosome? Yeah, people looked at that further, and they came up with this so-called CRISPR locus, where you have spacers from bacteriophages, and in between always the same complementary DNA. Yeah, certain fragments were always the same. And then people were wondering for a long time, okay, well, what's happening? What is that doing? Yeah, couple of questions around. Okay, so what then people figured out is that you have ways of adapting to attacks from bacteriophages. Yeah, what happens is that the bacterium is able to put some fragments from a bacteriophage into its own genome. Yeah, so it learns previous attacks. Now that you have this genetic information in your own genome, you can transcribe this CRISPR RNA molecules that contain again the modification. Yeah, this CRISPR RNA is loaded by the Cas9 surveillance complex, and then that one diffuses around the cell looking for invaders. Yeah, so if you now the same bacteriophage, same kind of bacteriophage comes, tries to put genetic information into the cell to turn that into a factory of viruses, then as long as the Cas9 with the complementary strand finds that in time, it can start cutting this DNA into pieces. Yeah, so it's a very interesting biological mechanism where, again, you take some DNA from the bacteriophages, you put it into your own chromosome, and then you have a defense mechanism that allows you to fight off attacks if this occurs from the same bacteriophages. And the question we asked is, how fast can one of those molecules find a target? How long does it take? Yeah, so why does it matter? Because if Cas9 target search is too slow, that would mean that I turn the bacterial cell into a factor of bacteriophages, and the cell would obviously die. Yeah, so you have to be there in time. Okay, how do we do that? So the work was published 2 years ago, Cas9 together with our microscopy framework that I was talking earlier about. And we have a similar story where we looked at the native CRISPR target search of a multidimeric protein. So this is the cascade, but we will focus on this Cas9 because it's relatively simple. Okay, now what's the issue? You have now, you want to target a genomic sequence from a bacteriophage, but you have the same sequence on your own chromosome. So how do you make sure that you not accidentally start cutting your own chromosome into pieces? Yeah, so this is why there is a fail-safe mechanism, and it's kind of like a two-step verification system that people nowadays use for security purposes. What's the idea? So you load your guide RNA, and there is a small 3-nucleotide sequence on the chromosome where first the protein tries to scan for that sequence. So it basically first it looks for NGGs. And only if it finds an NGG, yeah, so GG here stands for, so N can be anything, GG is specific for Cas9, only then it checks whether there's complementarity to the target or not. Because the initially, yeah, in that one, this piece of DNA was selected because it contained an NGG in the surrounding. But by building it in, it prevented to have, so here is no NGG present next to it, next to the spacer. So that means the selection is based on the NGG, on the bacteriophage, but in the own chromosome, this is not present. Yeah, so that means if there's no NGG, it doesn't matter whether I have complementarity to the target or not. Yeah. Okay, then the question for us was now, how fast is that screening? Because it kind of represents a bottleneck. You're not primarily looking for a target, but you're first looking for this relatively small sequence. And if you now do the maths and think about, okay, on your chromosome of millions of base pairs, how many NGGs do you have? A lot, few 100,000. Yeah, so you have to check basically a few 100,000 NGGs to check then whether you have complementarity to the target. And there was kind of an estimate that it is relatively fast, so smaller than 30 milliseconds, but it was not clear how fast it was. So what we now did is we labeled this Cas9 with a photoactivatable fluorescent protein. Yeah, so in order to kind of track the movement of the cell, of the protein in the cell. Why do we use photoactivated localization microscopy? Yeah, so in the normal case of a fluorescent protein, it's fluorescent, you're excited, it emits light until it photopleaches. But if I have a few hundred proteins in a cell that are fluorescently labeled, then I wouldn't see anything because they all overlap, because they are all fluorescent at the same time. So by doing a photoactivation step, I can tune the laser intensity of this ultraviolet laser such as that on average, I have less than one protein per cell that is actually fluorescence. Yeah, so all the others are dark. Okay. Moreover, and I hope we'll hear something about that later as well, the photoactivation enables counting, because I can now count, okay, how many molecules did I actually turn on per cell? And that's going to be important. Okay, how does it look like? First, some overview. So again, what I've shown you earlier, this is a bright field images of cells. So on the left side, we didn't induce any Cas. On the right side, we have a high Cas induction. So we have basically many of those molecules around. Then we excite with a laser, and you see it's all dark. We are happy. And then I will show you a video where at one point we switch on with the UV laser to photoactivate them, but we use a relatively high laser intensity. So let's see whether this plays. Yeah, here it goes. Yeah, so suddenly all of them turn on. And you see that every cell is loaded with a lot of Cas9 molecules, which you would like to have. Then, of course, they bleach. And if you look very carefully, you see that at the end, when you only have a few present, they wobble around a bit. Okay, so if you have a low induction and low photoactivation, we can find conditions where we basically see a single one moving around. Yeah, so this is this kind of white thing. So we added cell outlines in green, the track in yellow, and that basically moves around the cell. That's the longest track we can find because normally it will bleach after a few minutes. Okay, so how do we analyze that? That's now classical physics in a way, Brownian motion. So we calculate the mean square displacement. So let's say we have 5 localizations, 1, 2, 3, 4, 5. Then we can calculate the mean square displacement by looking at the distances between those. Yeah, so we have 4 distances of 1 time step, 3 differences of 2 time steps. And by fitting a curve through that, we can determine the diffusion coefficient as a hint on the mobility per molecule. Yeah, so we have a track, couple of localizations, and we have a mobility per molecule. Okay, now what did we do? Yeah, so we added 2 plasmids. Yeah, so one with the Cas9 and the PAM cherry, another plasmid now with a dummy plasmid without any DNA target sites. And what we saw is here we plotted a couple of tracks for different cells. You have a couple of cells where you see only a few tracks, and you have some cells where you see hundreds of tracks. Yeah, so you have very different copy numbers. And we kind of analyzed the mobility, and the prediction is now the following, that let's say you are bound on this PEM screening for a long time, yeah, then you have a relatively low mobility. If you are freely diffusing in a cell, you have a high mobility. Yeah, so if you plot a histogram of all those diffusion coefficients, you basically have 2 fractions: low mobility, high mobility. And if you now have tracks that change their status within the track, then you start seeing a merging of the peak. Yeah, because here you have either bound or non-bound. And here you have, okay, it diffuses for a few frames, then it binds for a short time and then releases again. Yeah, so the faster this interconnection will go, the more those histograms will merge, because we determine one diffusion coefficient for an entire track. And that basically provides then the information to calculate the transition rates. Yeah, what is the rate between those 2 states? And that is something we developed first in a Monte Carlo variant and then also with an analytical variant, so we can fit this distribution of diffusion coefficients, and we get out rates. Yeah, so 50 per second from this state to here, 43 per second from here to here. So then we asked, okay, does that depend on how many copies of the protein I have in a cell? Yeah, so we plotted the rates. We plotted the rates as a function of the copy number. Yeah, and more or less they are rather constant. Yeah, that makes a lot of sense because we have way more PEMs in a cell than we have Cas9 molecules. Yeah, so on average, it doesn't matter how many we have, rate is always pretty much constant. Okay. Wrap up, huh? Okay, now last bit is then what is happening if we add a target? Yeah, so now we put a couple of target sites on the other plasmids so that we can actually have an NGG where we can have a target bound molecule present. So we count, we have roughly 5 targets per plasmid, 70 plasmids per cell. We expect 300 targets, and that is roughly in line with the number of proteins that we have of the Cas9 in a cell. And that's the data. Yeah, so we have, first of all, we have this baseline of the PEM screening, yeah, freely diffusing PEM screening. That's always there. That's the green bit. And now we have an additional fraction that reports on the target bound molecules. Yeah, so in this case, if we have a low number of Cas9 molecules, we see up to 30% of the proteins are in a target bound, relatively stable state with low diffusion. If we look at high copy numbers, then this probability of being target bound decreases. Yeah, so we have then only 10% of the molecules that are bound. Yeah, if we plot that more carefully, then we see that we see a decrease of the target bound as a function of the copy number. That makes sense because we are starting to saturate all our target sites. If we assume that we have 300 target sites, 1,000 molecules, then they cannot be bound all the time. So the surprising thing is that we do see only 30%, even if we have a low number of Cas9. So what does that mean? That means that you need to have in your system an off rate, yeah, to basically go back to that situation. And we determine the off rate to be on the scale of 1 per every two minutes, yeah. And since we now have all the information available of all the rates, we know how many targets we have in a cell, we know how many proteins we have in a cell, we can now make a prediction to plot what is the cleavage probability or the probability to find my target as a function of time. Yeah, if we take out, let's say we have 1 unit present over here, then we see a 50% probability to find our target after roughly 4 hours, 240 minutes. Yeah, so that's the information that we basically got out, that for 1 Cas9 molecules takes around 4 hours to take a target. If you have 10 molecules or 10 Cas9 molecules present, that goes basically back to 20 minutes. And then this is the way how to analyze it. That's it. So thank you very much for your attention. So I hope I make you aware that open science is a good idea. Yeah, please do it if you can. The single molecule localization microscopy allows bridging structural biology and observing cells in real time. And we use that kind of to quantify target search mechanisms of CRISPR-Cas. And thank you for your attention.
Thank you very much, Johannes, for the nice talk. We have time for some questions.
Thank you. That was super interesting. I was wondering, don't bacteria also have, in addition to pure PAMs, also when you don't introduce an actual target, sequences that are close to a target sequence? So don't those also affect your analysis, and in which way?
Yes, yes. That's a problem. Yeah. So in our model, we simply ignore it. Yeah, we say simple, okay, this is a 2- or 3-stage system. I mean, people, especially with DNA curtains, yeah, have shown that you always have a certain probability of off-target binding. And yeah, that makes it then very complicated.
Does it affect your off rate?
Probably yes. So we didn't quantify it because it just gets too complex. I mean, there's one way around that I can potentially see that if you use kind of technologies like MinFlux, that you track it really one by one and see really how it binds to a certain spot of DNA. But in the end, you have so much DNA present, yeah, so it always will have an affinity to something somewhere. Yeah.
Yeah, thank you for the talk. You showed us how to resolve a big blob that's consisting of many marked proteins, right? By going to a state where only few of them light up. And then you said you have to wait to actually get all the information where the individuals are. But how do you make sure that they are not moving around while you wait and that your information is disturbed by that?
Depends what you want to know. Yeah, so in our case on the Cas9, we obviously want them to move because that's exactly the information that we want to analyze. Yeah, if you want to be really structural features, then you would have to make sure that they do not move. Yeah, but normally everything moves in a cell anyways. Yeah.
But what would be like a way to fix them?
You can fix cells, yes. Yeah. So if you're just interested in the localization, but not in the dynamics, then you could fix the cells, basically pin down everything and look where things are. People have done that to, for example, look at RNA polymerase complexes, where they are in the cell clustering and so on. Yeah, fixation is then the way to go.
Thank you very much for the talk and also for sharing it all in open source. I really like the spectral resolution single molecule. And so you have to search for pairs.
Yeah.
and they come with this defined distance. Yeah.
Yeah.
But there's also the condition that they are temporal correlated. Do you take this as a control that they
Yeah, of
must
course.
disappear at the same time? Because spatial alone could lead to some misleading interpretations.
It depends at the end on the densities that you measure at. Yeah, if you keep the densities in the blinking such as that you have enough space in between, then our approach works pretty well. I mean, this of course could be another way of sorting molecules. So now we look frame by frame whether we have a pair.
Okay.
Yeah, so if we cannot find a pair, then we discard it. It is surprisingly robust. There's also, if you're interested in that, in the paper somewhere in the supplement, there's a very nice analysis how to do that with Fourier transformation of a Fourier transformation to get kind of the estimate for the spacing that you then.
And you showed that you can do FRET using this technique.
Yeah.
Can you do this also inside cells? Was this in the paper?
We haven't tried that. I mean, I know that my former postdoc supervisor, they kind of electroporated double-stranded DNA into bacteria. Technically should be possible, but you need to have a FRET sample in a cell. It's not trivial.
Johannes, thank you for the talk. I was wondering about your phaser approach for the 3D SMLM. Is it only limited to the astigmatism-based approach, or could you also, for example, use it for point spread function engineering, for example, double helix?
Good that you ask. We have another paper on that where we looked in exactly into that.
Okay,
Yeah,
so it works.
I mean, again, the limitation of phaser is that you always need some kind of symmetry. Yeah, so in very weird things where you don't have symmetry, that's not the best approach. If we have a lot of optical aberrations, I didn't talk about it, then we can kind of correct for that with adaptive optics. You need symmetry. Yeah, if you have symmetry, then we analyzed tetrapod and subtle point and all those things. That kind of works. Is not yet implemented into ImageJ because I have no one doing that.
Okay, thank you.
But it's available for MATLAB.
Johannes, how many different FRET states can you discriminate? So how good is your spectral resolution for this? Will it replace the dual view or not?
Yeah, I mean, if you can dual view, if you have a dual view, use the dual view. Yeah, because then you have just a green and a red channel. Fine. Where our technique is good is if you want to do, let's say, 3-color, 4-color FRET. Then with a dual view, you always split into different regions, which I don't need to do. Yeah, also if you expect spectral changes, because there are some, for example, Nile red fluorophore is sensitive on the hydrophobicity of the environment, where you would like to see then changes within, then that's great. Complementary techniques, I'd say.
Yeah, thank you for your talk. Regarding the spectral resolved localization, so I mean it's a trade-off of your localization precision and the spectral information.
Yeah.
So how much of your localization precision you sacrifice to distinguish for, let's say, Psi5 and Alexa 647, which are spectrally very similar but shifted by 10 nanometers?
Yeah, so back on the envelope calculation is if your transmission grading has an efficiency of say 60–70%, and then you split pretty much your photons into two streams, so 0th order, 1st order. So let's say you have 30–40% of the photons available that you would otherwise use for localization. Yeah, so localization precision scales with 1 over the square root of n. Trade-off is not too bad.